Okay, so with this activity, again, I want to emphasize that you can submit this, resubmit it, and get an improved grade. I know some of you came in late, some of you had other issues, some of you were absent. So submitting it after you've had a chance to review this video and make any necessary corrections, it would be fine. All right, so the first step here is to go to this source, uh, this website, and draw a concept map that identifies the various cellular signals and messages involved in the pancreas. Now, some of you, which is fine, you use the illustrations. They are found on the website. That's fine. But I guess the key here is a less refined answer and a more kind of schematic answer. So you can go down the rabbit hole really quickly with this material. You could get into some really detailed work, and unfortunately that would be not as constructive as understanding the general outline of the process. So you have a pancreas, and I'm just representing the pancreas here as a square. And in that pancreas, you have these receptors that measure how much blood glucose you have. The one for the alpha cells. The alpha cells have these, and the beta cells have these in the pancreas. There's also delta cells that can measure that are found in that produce hormones that inhibit both alpha and beta cells. And then you have a third cell in the pancreas some of these cells are found in other organs as well, but in the pancreas they're found. And these receptors and cells produce proteins that inhibit or reduce consumption, appetite. So the PP cells produce pancreatic polypeptide, which decreases appetite. The delta cells, they produce a hormone that then feeds back and inhibits the alpha and beta cells from producing both insulin and glucagon. When the blood glucose is high, the alpha cells are stimulated via receptor, or the beta cells are stimulated via a receptor. And when that high glucose level is received, that message is received via this receptor, again, we could label the receptors, we can get in all kinds of details. I don't think that's very useful. I think it's important to know the beta cells. I'm trying to focus here on what's important for you short-term and long-term, that the beta cells produce insulin and therefore have to be able to measure when blood glucose is high. And the alpha cells produce glucagon and therefore have to be able to measure when blood glucose is low. So when the blood glucose is low, that's dangerous. You can go into a coma. So we have to make sure that that doesn't happen. So you have blood vessels. Obviously, blood vessels innervate the entire organ. Let me see here. Blood vessels innervate the entire organ, but I'm just drawing this simple, again, trying to keep the schematic simple to get the idea across. So as the blood comes by, the pancreas measures the blood glucose via these two receptors, the beta receptor and the alpha receptor, the receptor on the beta cells, the receptor on the alpha cells. If the receptor on the beta cells senses high glucose, it releases a hormone, the hormone insulin. Insulin's a protein hormone, polypeptide hormone. And if the blood glucose measures low blood glucose, if the pancreas measures low blood glucose to the alpha receptor, then it's going to release a hormone called glucagon. Glucagon has a series of effects, and insulin has another series of effects, and they're opposite one another. So now we're going to deal with, now we have to consider a feedback mechanism here. So remember what we talked about when we talked about feedback. So glucagon is going to be released, and it's going to have a series of impacts. And the series of events are pretty simple. The glucose goes down, which leads to increased glucagon as glucose in the blood level goes down. As you don't eat, you go through periods of fasting. Glucose goes down, glucagon goes up. That releases your glycogen that you have stored in your liver. This glycogen is stored in your liver, and we've mentioned that previously as well. In your liver, you have stored glycogen. Glycogen gets broken down. Remember, glycogen is a polysaccharide. It's a stored form in animals, very much like starch is stored in plants, and it's a branch form. We're not going to get into details. I don't think it's very useful to do so, but the bottom line is that you have this polymer, this really long branched form of polymer of carbohydrates, of glucose, that we call glycogen. That's where one of the functions of your liver is to store that glucose for times of fasting. That glucagon tells the liver, break down the glucose and release it into the blood. That increased blood glucose then decreases glucagon. So the decrease in glucose increases glucagon, which then releases glucose, which increases glucose in the blood, increased glucose, blood glucose, decreases glucagon. So this is a kind of a flow chart that shows negative feedback. When we say negative feedback, that means you're going to have the opposite reaction. As blood glucose goes down, glucagon goes up. As glucagon goes up, glucose is released, so glucose goes up, so glucagon goes down. So they have an opposite effect on each other. That's negative feedback. That's one of the primary ways your body is able to regulate most of its functions. Negative feedback is very much a regulatory pathway for all living systems. So negative feedback balances or creates homeostasis. That means you're keeping blood glucose about the same. Now, that's not enough to keep blood glucose stable. That helps, as you can release stored glucose or stop releasing stored glucose. That's important. But you need help to do that, so there's a secondary hormone, a second set of cells, as we've said a minute ago. We have these cells called the beta cells. This, of course, is happening with the alpha cells. The beta cells have another pathway, another flow chart. As glucose goes up, again, above some standard, so let's just go ahead and draw a line. Let's say there's this normal, let's call this normal, meaning average blood glucose. That's not dangerous, it's healthy. Obviously, your blood glucose is going to go up when you eat and down when you fast and back up when you eat, etc., back and forth around some normal range. This is what we would call a normal range. So, oops, let me use a green color. This is a normal range of blood glucose. The beta cells are going to measure increased. So their receptors are going to say, oh, increased glucose. Blood glucose is getting too high. And we're talking about blood glucose. We're talking about an organ in a pancreas, surrounded, innervated by capillaries, arteries, and veins. And the receptors easily comes into contact with blood glucose. And increased blood glucose leads the beta cells to produce insulin. Insulin, in turn, let me go ahead and take a look at this diagram here. In every body cell, every one of your body cells except for your brain cell, your red blood cell, your liver cells. Sorry, I had to write these with my finger because my eye pencil went dead. Your kidney cells and the lining of your small intestine, all of them, except for these tissues, all of them respond to insulin and glucagon. These particular tissue cells do not need insulin or glucagon to absorb sugar. They don't need insulin to absorb sugar, and glucagon has very little impact on them. Now, that makes sense if you think about it. Your brain needs to get sugar as fast as it can. If it doesn't have enough sugar, you faint or go into a coma. So making sure your brain has enough sugar is a priority for your body. So insulin and glucagon are not necessary. This is the one big drain in your on your sugar supply that keeps your brain, and so you need to make sure that you're not regulating this too stiffly. Red blood cells, mature red blood cells, don't have a nucleus, and so they're not going to be able to produce a lot of proteins. They don't have DNA, so they don't really have mRNA after they're mature, readily available. And at least they can't make any more mRNA because they don't have DNA, because they don't have a nucleus anymore. All that's been gotten rid of in a mature red blood cell in order to make room for the hemoglobin. So it still has mitochondria. It still can process energy. So it needs the sugar, but it can't make the receptors. It can't make the channels. The glucose channels are sitting in most of your body cells in the cell, waiting to receive the signal to be released and be incorporated into the outer membrane, the plasma membrane. The liver does a lot of that functioning, releases the glucagon, makes glucagon, so it makes sense that the liver do all the different processes it does not need the insulin or glucagon receptor. The lining of the small intestine, the small intestine is absorbing a lot of the sugar from the food that you eat, so it makes sense that you really want to let these things absorb as much of the sugar as it can, as quickly as it can. And your kidney is doing a lot of functioning and has to process sugar, is processing all your blood and filtering all your blood. So again, it makes sense that this insulin glucagon regulation is not happening in these body cells. But in every other one of your body cells, you have both glucagon receptors, or at least you have insulin receptors in all your body cells. Glucagon receptors are mostly found in the liver. So with insulin, once the glucagon cells, once the alpha cells receive the beta cells, the beta cells receive the high sugar signal on their receptor, they release insulin. So they release insulin. Insulin goes around and signals a body cell. There it is. So the insulin cell binds to the insulin receptor. The insulin hormone binds to the insulin receptor. It's a polypeptide hormone, so it does not need to be carried in any kind of vesicle, and it binds to. So it's water soluble. To be clear, the polypeptide tends to be water soluble, so it's floating in the blood. It gets distributed in the blood supply directly, and once the insulin gets to high enough level, it binds to the receptor. That receptor tells the cell to through a secondary messenger. So it sends the insulin binds, the insulin is the primary messenger. Then there's a secondary messenger within the cell. We'll talk about it a little later. They then tells the vesicles holding the glucose channels to release. Then those glucose channels then release and get embedded in the membrane, allowing then glucose to come into the cell. And you have to imagine this is happening 1,000 times what I'm drawing here. So glucose is able to get into the cell now. Now, someone asked about secondary messengers. The primary messenger here is insulin, where the secondary messenger, in this case, we'll talk about later, is actually getting is having a response inside the cell occur, releasing the channels that have been stored, also stimulating the DNA to produce more of these glucose channels. When the blood glucose that's when blood glucose is too high, allowing the glucose to leave the blood and go into the cells, bringing down your blood glucose. Remember, we're talking about your blood. We're talking about arteries and veins and the surrounding and capillaries and the surrounding cells, the surrounding tissues. When this glucose is high, the glucose goes into the cells because of the insulin receptors. The insulin receptors signal a secondary messenger, releases the glucose channels, allowing glucose then to enter the cells and leave the blood. So blood glucose goes from high to low. So now blood glucose is lower. So blood glucose high releases insulin. Insulin then signals his body cells to release the glucose channels. Channel proteins. These are proteins already made that allow glucose into the cell. Those channel proteins embed in the plasma membrane, which then allows glucose to enter which then lowers blood glucose, right? So blood glucose is high, it increases high blood glucose, stimulates insulin release, which lowers blood glucose. So this again is a negative feedback. High blood glucose, increased insulin, lower blood glucose. So increased insulin leads to lower blood glucose. So to simplify the flow chart here, increased insulin leads to decrease in blood glucose. A negative feedback. Remember, negative feedback means the opposite. When you have an inverse relationship between the two variables. So when you're dealing with negative feedback that allows the body to regulate things, not allow things to get too high or too low. So those are the beta cells in your pancreas. Those would be the flow charts that you can draw. And these are some of the schematics and exceptions. What part of this would you need to know moving forward? Well, I would definitely know the relationship between glucose and glucagon and what it does, that it increases glycogen breakdown, glucagon increases, glucose is too low, so it increases glucagon breakdown and the biochemical pathways that allow for glucose to be made in the blood, therefore increasing blood glucose, right? And that that's done by alpha cells, right? So alpha cells deal with the glucagon. The beta cells of the pancreas then create insulin, and they react to high blood glucose because of a receptor that measures blood glucose level. High blood glucose stimulates insulin production, and that means the DNA is stimulated, and you start to make insulin. Insulin then, through the pathway, what pathway would we be talking about? We'd be talking about transcription, then we'd go into translation. The translation would happen in the rough endoplasmic reticulum, because insulin is being secreted by these cells. So they're leaving the cell. The pathway for Proteins that leave the cell go through the endoplasmic reticulum, rough, then the smooth. They're changed and packaged and into the Golgi body, from the Golgi body into vesicles, out of this of the beta cell plasma membrane into the blood. That blood insulin then goes around, attaches to receptors at the body cells that then stimulate the embedding of release of glucose channels, which then embed into the plasma membrane and allows glucose to enter the cell, therefore leave the blood. Right? Entering the cell means it's leaving the blood, so increased insulin decreases blood glucose. Again, we call this a negative feedback. So I'm going to leave this video here, and I'll deal with the rest of this packet in a secondary video. I just thought this would be enough for you for now, and we're like at 20 minutes. So I hope this is enough explanation for this kind of schematic drawing. When I ask you to do a flow chart, you can see this is a flow of events. All right.